So we started talking about growth last time, right? And we ended with the important things for growth, right? Having the correct temperature, the correct oxygen level, pH, and water availability. And again, as you think about, and especially when we get to our talking about control, one of the new books that I'm looking at for the other microbiology course, I love it. They put growth and control in the same chapter. I'm like, that makes sense, right? Because they're interrelated, right? In my other class too, I put two of the parts together. I put it together as growth and control. I don't even separate them. So because what is needed for growth can be taken away to control growth, right? Or can be tinkered with. So how do we use temperature to control the growth of microorganisms? We increase it, right? And what is that? What do we call that? We cook our food, right? It's cooking. That's actually one of the main reasons why we cook our food is to kill microorganisms, right? So everybody knows about hamburger meat, right? It comes pre-contaminated, y'all, most of the time. It already has bacteria in it. This is why you should cook it to the temperature they say to cook it to, because it will kill the organisms that are most commonly found in that meat. And that's why the different temperature ranges for the different meats, right? Because it's to kill the microorganisms that are pathogenic, that could be and are usually associated with that particular food product. That's the why, right? So what else do we do? So we heat to kill. What else do we do to control microbial growth as it relates to temperature? We refrigerate and we freeze. I got news for you. Microorganisms, you can freeze them and they will not die. That's how we long-term store the bacteria for the microbiology lab. They're in my freezer. Well, the microbiology freezer, not mine. I have brought stuff home, but not bacteria. Gotta love the lack of power we sometimes have here. Proteins, right, as we've said before, don't like to be at unstable temperatures, right? And that's really what's important here. And this is what you're messing with temperature, is protein structure. So you can destroy that with heat. But you pretty much just, as the name says, you just kind of freeze it. You just put it in stasis when you freeze microorganisms. So they're not actively growing. They're not dead either, usually. Some of them will die, but most of them will survive the freezing process. So when you take that hamburger meat back out of the freezer, once it gets back up to being warm again, they'll start growing again, right? So don't leave it out on the countertop all day to defrost. Probably not a good idea, right? And then, so refrigeration, again, is going to slow down most microorganisms that we're concerned with, right? It's going to slow them down. It's not going to stop them. Oxygen. What are some very popular things that we do nowadays to remove oxygen from our foods to help preserve them? They got special little machines, but they even got little pumps that you can put. Not dehydration deals with water, right? So that's how we limit water availability, right? We're removing water by dehydrating. All those vacuum seal systems, right? What are we sucking out? Air. What's in the air? Oxygen, right? There's a problem with that, though. Do all microorganisms need oxygen to grow? No. For some of them, you just made them very happy, right? For others, right, you did prevent their growth and you may even kill them if you're able to eliminate all the oxygen from the container. So sometimes we combine these methods, right? So coffee is vacuum sealed and a lot of times freeze-dried, right? So they're removing moisture, right, and removing air. They usually do both, and sometimes even freeze it. This is done with medications as well sometimes. And then pH, this is something that's been used for a very long time to help preserve foods. So what in your house is preserved by a change in pH? Anyone know? Who likes pickles? Sauerkraut, right? All these fermented foods, right? They contain a very high concentration of acid. The most common acid is vinegar, right? And that very high acidic content, very few bacteria that are pathogenic to us can grow at that high acidic level. So in that process of making the acid, sometimes of fermenting those foods, things like kimchi and sauerkraut, is actually microorganisms that cause that process to happen. But again, they're not ones that are going to make us sick, right? But they're going to prevent the other guys from growing. So when we talk about temperature, scientists love to categorize things, right? So we have 5 distinct categories, classifications we do for organisms based on what their optimum growth temperature is. What's the best for them? And if they fit within this range, they fit within that category. So unfortunately, their categories are based on the Celsius scale, right? And most of us are familiar with Fahrenheit. So the Celsius scale is based on the properties of water. So at 0 degrees Celsius, water freezes. What happens to it at 100 degrees Celsius? It boils. It boils, which is 212 degrees Fahrenheit, right? And we all know freezing, which thankfully we don't experience too often in New Orleans, is 32 degrees Fahrenheit. Your refrigerator is typically around 4 degrees Celsius. Room temperature of about 70 degrees Fahrenheit is around 20 degrees Celsius. And then body temperature of 98 degrees Fahrenheit is 37 degrees Celsius. So you'll notice in the microbiology lab, we have an incubator set at 37 degrees Celsius. Right? That's the most commonly used incubator because a lot of the organisms we work with are pathogens, which means they would like to be living inside of us. Right? So being at our body temperature makes sense to them. So also, if you notice the categorization, right, the category associated with human body temperature is known as mesophiles, right? And notice it's right there in the middle, right? That's us, right? We're mesophiles, if you were to put us in this classification system. And then you have the much higher temperatures, right? You're talking about right at boiling, right, for water. And thermophiles, just to give you an idea of how hot around 70 degrees Celsius is, it's 158 degrees Fahrenheit. So that's pretty darn hot. So where would you find these type of environments? So there are microorganisms that live in all these types of environments. Now when you get to the really extremes, what domain of organisms are we probably talking about? Archaea, right? Those extremists. Especially when you get to the really cold and the really hot. So the very coldest range, right, where we're talking about just above freezing for water, things can grow. It has to be above freezing because freezing, water is frozen, is solid, is no longer available for use by an organism. But just above freezing, pretty darn cold, right? It's still liquid. It still can be used for growth. So those are your psychophiles. So remember, phile means what? To love, right? To love. And psychro actually means cold. And as far as I'm concerned, if you like the cold, you are crazy, right? Psycho, crazy, right? I'm not a big fan. So where on earth would you find this really cold environment? In the Arctic, right? So we're talking, glacial lakes. Places like that. There are algae that grow associated with, so we're even talking about eukaryotic organisms here. Algae that grow associated with the glaciers in Alaska, right? And their spores contribute to the different color you'll see sometimes in the snow and associated with the glaciers there. You'll see these really pretty pink and blue ribbons. Those are actually microorganisms growing in that environment. Psychotrophs, right, our next level, so a little bit warmer. We're talking about, more happy at room temperature, but can grow at refrigerator temperatures. So notice at these lower temperatures, some of these organisms can grow there. So this grouping of organisms we really worry about being found in our refrigerated foods. Mesophiles, right? As we talk about microorganisms, these are going to be the ones that make us sick, right? These are going to be the pathogens that make us sick. So these are going to be ones found in the human body are going to be mesophiles. Do we necessarily need to worry about psychotrophs? And I'll back up for a minute so you can see the humps a little bit better. So this is, they're barely growing. This is they're super happy, right, around room temperature. And then if you get higher than that, which is right here at body temperature, they're going to die off. It's too what? Too hot. You're going to denature their proteins. So those ones that are growing in our refrigerator, some of them aren't going to grow in us. The problem is that they produce toxins that then you ingest, right? And that could make you sick. So microbial growth isn't always what makes us sick, right? And we'll talk about this in our next unit. A lot of microorganisms produce toxins, and that's what can be the worst part of that infection, especially with tetanus, right? It's really the toxin we're terrified of, not necessarily the bacteria. But the problem is the bacteria makes the toxin, right? Makes the bad stuff. So as it relates to food, too, what's another toxin producer that we worry about? It's another Clostridium. Causes botulism, right? So again, it is the toxin that makes us sick that the bacteria produces. The bacteria, for the most part, doesn't grow in our bodies. The problem is it grows in your food and then produces that toxin, and then you eat that toxin, and that's what makes you sick. So thermophiles. So again, we're talking about really hot temperatures, right? These guys you'll find in the hot springs in Yellowstone National Park, right? That first picture I showed you guys last time. In the center of your compost heap, right? That'll get really hot. The microorganisms that are decomposing that organic matter. Again, do you have to worry about any of these making you sick? Well, I don't know about you, but I'm not drinking or hopping in a hot spring. Some of those are too hot there, right? We don't really want to go above 100 degrees Fahrenheit, right? Your hot tub, right, is not in the classification of thermophile environments, okay? Hot tubs are usually around 100 degrees, right? We're talking about, not boiling, but pretty darn hot will burn you water or environments like the compost heap. But I don't know of any organisms in those types of environments that are pathogenic to us. But again, we're not usually exposing ourselves to stuff from those types of environments. Hyperthermophiles would be found in hydrothermal vents. So in aquatic environments where magma is actually coming out into the ocean, right, where it's extremely hot, the water is just under the boiling point. There have actually been microorganisms found growing there. And then, of course, some of the hot springs are that hot in Yellowstone National Park, so some of those would even be classified as hyperthermophiles. You see the steam coming off the water. It's Pretty cool place. But these guys, the thermophiles and the hypothermophiles, are why we have PCR today, polymerase chain reaction, where we can actually have enzymes in a test tube, in a machine, and it actually copy DNA for us, because the enzymes that we use to copy the DNA come from these organisms. So we can use heat to separate the strands of the DNA and not denature those proteins in the process. So the strands separate, the enzymes attach, they copy the information, then we heat it up again and split it. All the while, the enzyme is not destroyed. If we were to use our own enzymes, if we heat it up, we destroy the enzymes, and you'd have to keep adding it in each cycle. It just wouldn't be productive. There wouldn't be any point. So those organisms really have to have major changes to their proteins to be able to survive at those really high temperatures. So the next we're going to look at is oxygen requirements. So we already know, right, some organisms like ourselves absolutely require oxygen. Others can grow without oxygen. So we have several categories that we need to be able to define. And we need to be able to classify the bacteria based on the basis of their oxygen utilization intolerance. So oxygen actually can be toxic, right? So you have to be able to tolerate it, to live in the presence of it, and maybe even to use it for your metabolism. And then we'll look at the effect of water availability on growth. So for obligate aerobes, these guys absolutely require oxygen for growth. Obligate means you have to. Sometimes it will also be referred to as strict aerobes. And that's what we are, right? Without oxygen, we're dead. Huh? Strict. Strict. So obligate anaerobes, these guys, again, no oxygen. So and this means without, right? So without oxygen. So this is the case for Clostridium botuli or Clostridium tetani that causes tetanus and the one that causes botulism. These are actually anaerobes. They will not grow in the presence of oxygen. But they're both spore formers. So they both can protect themselves when oxygen is present. But when oxygen is gone, they're going to start growing under those anaerobic conditions. So when we can foods, right, we suck the air out, do we not? You create an anaerobic environment. There's always the saying, beware of the swollen can, right? Something grew inside of there. Don't mess with it. Throw it out. Dented cans are all right as long as they're not punctured. It just means somebody dropped it. It's the swollen cans you don't want. I've actually seen one once, cleaning out Grandma's shed after she died. She had a stockpile of canned goods, and there was one very swollen can. Had I known I was going to be a teacher back then, I probably would have took a picture so y'all could see it. But it does happen. Occasionally it will happen. But the canning industry, has strict controls on that for commercially canned foods to make sure that Clostridium spores for botulism don't get into the food products. Facultative anaerobes. These guys prefer an oxygen environment, but as their name implies, anaerobe, they can grow anaerobically, typically via fermentation, which does not require oxygen. They don't grow as fast when they ferment, but they'll still grow. They still can survive. Facultative meaning the ability to change, right? So these guys can grow with oxygen and will grow really fast. Escherichia coli is a facultative anaerobe. A lot of our gut microbes are facultative anaerobes. They can live when the gut is anaerobic. They'll keep growing and they'll ferment. When there's enough oxygen, they're going to utilize that and grow even faster. Microaerophiles, micro means what? Small. So aero referring to air, philes, they love air, but they love small amounts of air. So these guys require very small concentrations of oxygen. They can't live without it, but too much will kill them. So our stomach, actually, believe it or not, is an environment where there's a small amount of oxygen. And an organism that can cause a really serious infection, known as Helicobacter pylori, is a microaerophile, right? It requires small amounts of oxygen. So the stomach is the perfect environment for it. That, and it's motile, it can swim right through the mucus layer in our stomach, and it can protect itself from the acids that our stomach produces, so it doesn't get killed by stomach acid. Unlike 99.9% of bacteria, usually a lot of them are going to get destroyed in your stomach, are going to get digested right along with your food, right, because of the hydrochloric acid and the protein digestion that happens in your stomach. Aerotolerant anaerobes. So as the name implies, again, they're anaerobes. They can grow without oxygen. Aerotolerant lets you know that these guys can tolerate oxygen. Even if oxygen is present, they're not going to use it, right? They don't use it, but they are protected from it. Because remember I said oxygen can be toxic. So you've got to have protection against it if you live in an aerobic environment, if you live in the presence of oxygen. So for us, for aerobes, we have two enzymes that help protect us. And you guys don't need to know them, right? So I don't have it written out for this class. But SOD stands for superoxide demutase. It's an enzyme that destroys oxygen-free radicals that form when you're in the presence of oxygen, especially if you're using the electron transport chain. And sometimes those extra electrons will get attached to oxygen and hydrogen won't get attached, and you won't get the formation of water. Instead, you get these really damaging forms of oxygen. So to protect ourselves, we have an enzyme that converts that oxygen radical into hydrogen peroxide. Hydrogen peroxide is then converted into water and gaseous, safe oxygen using the enzyme catalase. Right? So all of us that are aerobic, we all have the ability to break down hydrogen peroxide. So this is why when you pour hydrogen peroxide on a wound, right, it bubbles, because your catalase enzymes break down that hydrogen peroxide into water and gaseous oxygen, which is causing the bubbling. If it does not bubble, your hydrogen peroxide is old, throw it away and get some new stuff. Although they say for wound care nowadays, it's too destructive even to your own cells that they suggest that you don't use hydrogen peroxide on wounds. Soap and water and some Neosporin. But it's good for cleaning other things, right? And it can be actually used as a sterilant. It could be used to sterilize things in the vapor form. So, but organic matter contains catalase most of the time, and we'll break it down and make it not useful. So there's an old wives' tale out there. If you pour it on a wound and it doesn't bubble, it means it's not infected. It's a wise tale, right? It means your hydrogen peroxide is old and is mostly water. Throw it out and get some new stuff. It should always, always bubble. You produce catalase. So notice that all the ones that grow in an oxygen environment have at least superoxide demutase, right? So facultative anaerobes, remember we said they can grow in the presence of oxygen. Aerotolerant, as the name implies, they can tolerate oxygen. Why? They have that protective enzyme for the most damaging form of oxygen, that oxygen-free radical. What's the poor problem for these strict anaerobes? Oxygen will kill them. Why? They have no enzymatic protection. They don't have either one of the enzymes. So they strictly have to be in an anaerobic environment or they will be killed by oxygen. This is to our benefit, right? Because in this case, if we aerate things, we could kill them. But it's bad news, too, when we create those anaerobic environments, because then these guys can grow. And especially if it's a spore former like like the Clostridium tetani and botulism, you now just created an environment where they can grow. Those spores can germinate and form back the original cells they once were. So microaerophiles, again, they need just a little bit of oxygen. They have the best protection, and then their second level of protection is going to vary depending on the organism. So this growth pattern you see here are specially designed tubes that we use in the microbiology lab, where it creates an aerobic environment at the top, but an anaerobic environment at the bottom, where there's no oxygen. So if you have a strict aerobe, it's only going to grow in the oxygen zone. Where something like a facultative, it will grow in the oxygen zone, but it'll also grow in the non-oxygen zone. It'll ferment, but it's not going to grow as well, right? Its growth rate's going to be slower. That's why you see less dots, and you see more dots at the top. This guy tolerates oxygen. He's not really affected by it. It's not going to affect his growth, right? So you can see the growth is uniform throughout the tube, right? Because he's doing nothing but anaerobic or fermentation, respiration, and not utilizing the oxygen. But he can live where the oxygen is because he can tolerate it. He's got enzymatic protection. This guy, as we said, has to stay in that anaerobic zone or he'll die. No protection against oxygen. No tolerance to oxygen. And this guy, again, needs a little bit of oxygen, so he's got to stay where there's a low concentration of oxygen in the tube. That's why you see the growth not at the top, but just a little bit below the top of the tube. So, I guess pH is in here next. So pH, remember, is the partial pressure of hydrogen ions. So this is a proton, right? This is a hydrogen atom that's lost its electron. It is just a proton. And because of that, it can interact with molecules, specifically proteins. And this would cause the proteins to no longer work properly if too many hydrogen ions are present or if not enough are present. So in neutral situations, you have a balance. For organisms that we classify as neutrophiles, for them, they can go a little acidic, right? 5 would be considered acidic. 7 is neutral. And 8 would be basic, right? But they're right around neutral. They can survive in that range. For humans, though, our range is much, much more narrow, especially for our blood pH. It is 7.35 to 7.45. That has to be strictly maintained. If it's not, right, you will slip into a coma and die, or you won't be able to breathe and convulse and die. So it's strictly maintained. Microorganisms can exist and grow at 5.5 or lower pH. We refer to them as acidophiles, right? Because they can live in the acidic environment where there's lots of hydrogen ions. They do this by being able to pump hydrogen ions out of the cell to maintain a neutral pH inside that cell. So even though acidophiles, right, they live in an acidic environment, inside their cell they're going to still maintain mostly neutrality. But the problem is dealing with, sometimes when you bring stuff in, hydrogen ions take a free ride in the cell as well. And so they have to actively pump those hydrogen ions back out to keep the internal environment from becoming acidic and killing them. So some have adapted to those environments. Alkalinophiles, you have a problem now where there aren't really enough hydrogen ions in the environment. So to get them out of the environment, right, there isn't a lot. And so concentration-wise, if you're trying to bring more inside of the cell, the environment, that's not the flow, right? It goes from an area of high concentration to low. Hydrogen ions are going to tend to want to leave the cell instead of come into the cell. So in order to bring them in when the cell needs to, they've got to do ion exchange. They basically say, here, I will give you this other positively charged molecule. I'm going to give you some sodium. Please give me that hydrogen. I really need it, right? So they kind of do an exchange. And again, to maintain neutrality within the cell, though. For water, all organisms, Every single living organism, every single living cell must have water in order to grow. So if you don't have it, you could potentially die. But as it turns out for microorganisms, they're really good at surviving without water. They're not going to grow, but you're not necessarily going to kill them. And that's where this term that's sometimes used kind of drives me crazy, because it says plasmolysis. Lysis means what? To burst. But in this case, they do not. Okay? They in fact shrink. They kind of shrink up like a raisin. Right? You can stick a raisin in water, and it'll plump back up like that original grape it once was. Right? You can rehydrate it. You can do this with bacteria too. You can dehydrate them. You can remove water from the equation. They'll kind of shrivel up, but that cell wall kind of helps still protect them. And so when water becomes available, they'll plump right back up again. Now, some of them, of course, you can kill by removing water. But a lot of them can tolerate this lack of water. Some of them, too, think about it. What will make water less available in an environment? We salt things to help preserve things. Again, we're trying to get the water to come out of the product, out of the food, even out of the bacteria, because we have such a high solute concentration outside. We have a hypertonic solution, right? When we salt things. High sugar, right? That's why honey and syrup and things like that can be left out and not refrigerated, because very few microorganisms can grow under that osmotic pressure of something being so sugary, being so salty, right? When you cure meats and such as that. But if you add water back into the equation, right, they can usually revive from that. There are ones that are specially adapted, right, that are referred to as osmotolerant. And then there are even ones that have specialized to live in salt environments. So much so that some of them can only exist in high salt concentrations. And these are some of your Archaea, right, that are what we call halophiles. The halo refers to the high salt concentration. They absolutely love it. They've adapted so much that they can't survive without it.
I thought water follows electrolytes.
Yes, water will follow electrolytes.
But if you're just putting salt on the exterior portion of something to preserve it,
Mm-hmm.
then are you just getting the water to come outside?
Yeah. So when you salt food, right, you're drawing the water out of it. So the same happens to any microbes that are present there, right? If they're sensitive to this, not all are. And the bad news for us is Staphylococcus aureus, some strains of that, and most of the staph are salt tolerant. Because think about it, what part of our body do they typically inhabit? Where do you get staph infections? Your skin. Your sweat. Your skin is salty sometimes. So they would not be able to exist in that environment if they weren't salt tolerant, right? And some Staphylococcus aureus can produce toxins in our food, right? If you salted it, it still grows. It produces toxins. You eat it, you get sick, right? So that's one of the ones that they try to avoid, and that's why, even when we preserve foods, there are microorganisms out there that, depending on what method is used, it's not going to affect them, right? It can still get contaminated, still make you sick. And that's the scary thing. There isn't any one specific way to protect yourselves, right? That's 100% foolproof for most things. Just kind of frightening. So you'll notice in this lecture, I've got a bunch of examples of media that we use in the lab. And what I'm going to do Tuesday when you guys come to take your test, I'm going to actually have the real stuff laid out on one of the tables in the computer lab where we take our test, so you can actually see the real thing. What you need to know for the objective notice is to be able to define the terms related to the media. You don't need to memorize all these different types, but depending on your future careers, you may be exposed to some of this stuff, right? So I'm going to show you guys it. I'm going to talk about it, but by no means do I want you guys memorizing each one of the media and what they do. But more importantly, these different types of media and why we use them, right, are explained in their definitions. So we utilize complex media. We utilize chemically defined media, selective differential media. What are these? Why do we use them? So defined or synthetic media, this is media where we know exactly what the organisms are eating and in what concentrations. So I equate this to the Nutrisystem diet, right? You know exactly what you're getting. You're also paying for that, are you not? It's not cheap. We only have probably one media that falls into this classification in the microbiology lab, and that's only because we're using it for a particular test, not because we want to know exactly what the organisms are eating, that we want to basically put them on a Nutrisystem diet. These types of foods tend to be very expensive, just like Nutrisystem, because, again, everything is laid out. You know exactly what they're eating. For the type of work that we do in the microbiology lab, we don't need it to be that defined, right? We don't need to know exactly what they're eating. We just need them to eat and grow so we can play with them. So we use a lot of what's referred to as complex media. Where some, right, a complex media has at least one ingredient of unknown composition or concentration. So beef extract. Is all beef the same? No, when you go to the grocery store and you buy ground up hamburger meat, beef, right? You can buy 30% fat, 20% fat, 10% fat, right? And, you know, they've adjusted that for you. If you just, like, took it from the cow, it's going to vary from cow to cow, right? But do we care how much fat they're getting? No. We just want them to eat and grow. And obviously, right, they like eating us. Most microorganisms, right, that we work with are pathogenic. They like hamburgers too. Really, you know, all we are to them is a big fat juicy cheeseburger. Right? They're like, yum, when they see us. Okay? So we give them yummy fast food, right? We give them the fast food diet in the microbiology lab. Right? It's going to vary from batch to batch, right? But we really don't care. The main nutrients they need are there. We don't care how much fat they get. It's fine. Just grow. Make sense? So in a lot of microbiology labs, right, they use a lot of these types of media, right, where you have components like beef extract and partially digested proteins. Tryptome is where they use pancreatic enzymes and they digest the protein casein. Anyone know where casein comes from? Nope. Milk. It's the white protein in the milk you drink. Casein. Whey is the other protein in milk, commonly found in milk. Right. So, just like us, microorganisms like to eat it. I like this one, though. Pancreatic digest of animal tissue. Or peptic, excuse me. So, enzymes that digest proteins. They don't even tell you what animal it's come from. Right? That's definitely complex, right? We don't even know what animal it came from. Right? Why do we partially digest the food for them? Why would you do that? We just want them to what? Grow. Do we want them to grow slow? No, we want them to grow fast. So we're going to make the food readily available, already pre-broken down for them so they can easily absorb it and start putting it together to form their molecules and grow. Right? If you give a baby a carrot, he's not going to be so happy with you, right? He doesn't have teeth yet. He's going to sit there and gnaw on that poor carrot all freaking day. So what do? We grind it up for him. We make it easier for him to eat faster. We pre-digest it for the bacteria to make it easier for them to eat it faster. Right? If you don't have to chew, which is basically what we're doing for them, you will eat faster. Make sense? Okay. So peptones, right? Whenever you see those are protein hydrolysates, right? So we're partially digested various protein sources. Extracts or aqueous extracts, usually of beef or yeast, are the most common, right? Those two things are going to make a media complex, right? Because it's not defined exactly what is there, right? These are very varied protein sources. Agar is added to the food. This is a polysaccharide from marine algae that will solidify and stay solid up to about 60 degrees Celsius, which I forget what is Fahrenheit. Pretty darn hot. You have to basically almost boil it to liquefy it. Okay? And so that's what we do. We liquefy it, and then we pour it into tubes or petri dishes, and it will solidify. And we can stick it in the incubator at 37 degrees Celsius, 98 degrees Fahrenheit, and it won't melt. Right? So it gives us a nice solid surface for the microorganisms to grow on. And the good news for us, too, is that we haven't found a bacteria out there yet that eats it. So they don't digest it. They eat the food around it, embedded in it, but they don't eat the agar. So we keep that solid surface. And then the benefit, too, is that, it'll stay solid at those higher temperatures that most organisms we want to grow at. Gelatin, which we use to solidify things, right, is a protein. And as you know, it does not stay solid at room temperature, right? It starts melting. So definitely sticking in the incubator. And just like us, there are lots of bacteria that like to eat that protein, right? They'll digest it and eat it. And it'll definitely be goo then. It won't create any solidified surface. So supportive or general purpose medias, right? As I said, just to support the growth, right? We just want them to grow. So tryptosoy agar is very commonly used, nutrient agar, right? These are general purpose media used a lot in microbiology labs just to grow the organisms. Enriched media are a general media that usually something has been added. The most common enrichment is blood. So in the case of blood agar, whole red blood cells have been added to that. Right? And I'll show you guys one of those plates on Tuesday. Chocolate agar is not chocolate. Microbiologists have a sense of humor too. So guess what color it is? It's brown, so it looks like chocolate. But in fact, it's lysed red blood cells that are added to this media. And because they're lysed, right, you get that brown discolorization that happens to the hemoglobin mostly when you lyse the red blood cells. So.
This use most common because my mom uses this to hospital.
Yeah.
Is that because it's less similar to human or immune pathogens?
So red blood cells are a very rich source of iron, right? So a lot of microorganisms out there have adapted to going after our red blood cells to get iron because they need it too for their metabolism. So yeah, a lot of the tests that we run and the examples that I'm unfortunately not going to go over for time constraints, but you guys can read over. Strep throat is the best example of that. When I was a kid, you went into the doctor's office, they would swab your throat and literally rub it on a blood agar plate in front of you. A whole red blood cell blood agar plate. Because Streptococcus priogens, which causes strep throat, can literally lyse red blood cells. Just obliterate them. So when you look at that plate later, you'll see clear zones where they've completely destroyed the red blood cells. They're no longer there. Now they do the rapid strep test, but a lot of times they'll still do the swab, right, and send it off to the lab where your mom works, and she'll put it on the plates. So it depends on what pathogens they're looking for. But yeah, a lot of them really like red blood cells because, it's a rich source of food, especially iron. So selective media, when we utilize a selective media, we want a particular type of organism to grow. So the blood agar plates that we use in in the lab are also selective. They have an antibiotic in them that inhibits the growth of gram-negatives and therefore favors the growth and selects for the growth of gram-positives. A lot of your gram-positives are the ones that produce those hemolysins that can destroy red blood cells. In the case of McConkey agar, there are components within that agar that, again, inhibit the growth of gram-positives in this case, and therefore select for gram-negative growth. Right? We want gram-negatives to grow because we're looking for an attribute within those gram-negatives. So if a media is being selective, there's usually something toxic in it, right? Whether it be antibiotics or something else that's going to kill off a particular group of organisms and therefore select for the ones you're interested in, right? So that's how we're selecting. And selective media is used a lot, especially in clinical testing, right, when we really want to select for a microorganism. So this is an example of a selective media where it contains phenyl ethyl alcohol, right? And so this is really important to us, right? We use alcohol a lot as a disinfectant, especially if you use hand sanitizers, right? So what type of organisms is it typically affecting? So who's not growing on this plate? They streaked, but you see no growth here, Proteus vulgaris, and you see minimum growth here because there's a problem with alcohol, right? What's the problem with alcohol? Does it stay put? No, it evaporates, right? So this plate may have been incubated a little too long, and the E. coli started to come back. You see a little bit of growth. But E. coli and Proteus are both gram-negatives. Remember, gram-negatives have what as their outermost covering of their cell? LPS is part of that layer, but what do we call that layer? The outer membrane. It is a membrane, right? What is it made out of? Mostly. What are membranes made out of? Come on, y'all know this. The little head with the two little tails. Phospholipids, right? Remember that? Alcohol dissolves those. Remember in the gram stain, right? That outer membrane disappearing. That's what you're doing to these cells. You're mostly affecting gram-negatives when you're using alcohol, right? You're affecting their outer membrane. And that's how you're destroying or harming them. Gram-positives, on the other hand, no outer membrane. No problem with alcohol. You're not going to get these guys with that. So, again, there's no method for cleaning out there that's 100%. Right? But you get rid of a lot of things like E. coli, right, with alcohol. So McConkey that I mentioned earlier as being selective is actually a media that's also differential. It helps you differentiate one group of organisms from another. In this case, the ability to ferment lactose. So lactose is a sugar that's very commonly fermentable by microorganisms. This media contains components that selects for gram-negatives, and then when it ferments the lactose, it produces acids and interacts with the pH indicator and allows us to see by the pH change if that happened. So you'll see here that E. coli, see how the colonies are kind of pink? E. coli is a lactose fermenter. And it's gram-negative because it grew on these plates. Gram-positives are killed by the crystal violet and bile salts. These guys down here, Salmonella, it is gram-negative. It's growing on this plate, but you don't see the pink, right? It's not fermenting the lactose. So differential media help us distinguish different types of microorganisms based on their biochemical characteristics. So blood agar is an enriched but also a differential media. It lets us know if those organisms can lyse the red blood cells or not. And in fact, there's two different types of lysis that can happen. What we call beta and alpha. Beta I always refer to as bad. That's the one Streptococcus prurigens is. Completely obliterates your red blood cells. And I'll show you guys examples of that. McConkey, as I already said, we're looking for lactose fermentation. So again, what these media are, what they can do, not necessarily the specific examples are what you need to know, right? So up to this point is what you need to know, right? This other stuff is kind of bonus, so I'm going to skip it for now. And I'm just going to keep going with the lecture, and I'll just name it both. I don't feel like stopping it. All right. So control, the good thing about control is you guys know most of this stuff, right? We use this every day in our everyday lives. So as I've already said with growth, we use heat, right, to kill microorganisms. That's a physical method of control. Irradiation is being used more and more. Filtration. Mechanical removal. When you wash your dishes, right, you're mechanically removing materials. And then we use a lot of chemicals, right? A lot of chemicals, right? And those chemicals depend on the circumstances and the degree of control required, right? How clean do we need something to be? So we have some terminology that we need to know here, right? So we'll define those, and then we're going to differentiate the difference between antiseptics and disinfectants, which is one of the classifications that we're doing in our table, which I wanted to be done with yesterday, but it isn't done yet. So I may give you guys, I think, a little more time. So sterilization, what does that mean? It means removal of all microorganisms, but really we need to extend on that definition a little bit more because most scientists don't believe that viruses are organisms, right? But if something is truly sterile, you have to destroy the things that could make us sick, right? And viruses definitely can do that, right? As well as the dormant form of bacteria known as endospores. You can achieve sterilization through different methods. Some filtration methods, right, can render something sterile. You can sterilize air by filtering it. You can sterilize liquids by filtering them. Heat, right? There are different forms of heat. Some can lead to sterilization of liquids, objects. Chemicals and irradiation, some forms out there can render something sterile. You're not using any of these at home, trust me. You're not using any sterilient type cleaners at home. Okay, you don't need it at home. Disinfection. So this means we've eliminated most pathogens. We're worried about the stuff that makes us sick. So some viable microorganisms may still exist, right? So disinfectants, sometimes too, will kind of tell you what they specialize in. Biocides, right, meaning they're killing living organisms. Germicides usually includes all things that we consider germs. So we're talking about eliminating viruses and bacteria. Where bactericidal is specifically saying it is designed to kill bacteria, right? That's the focus of that cleaner. Disinfectants we use on inanimate objects and surfaces. Antiseptics, on the other hand, are safe enough to be used on living tissues. Can a disinfectant be an antiseptic? Yes. Can an antiseptic be a disinfectant? Yes. If it's safe to use on your skin, it's probably safe to put on something else you don't give a shit about. What you worry about is disinfectants, right? Are they too harsh to put on you? Right? So a lot of times a disinfectant cannot be put on your body. Right? And as we'll progress, right, and we're going to continue on this topic, right? Remember I said we're going to do a discussion board on it. So my big fat Greek wedding, what does he use as an antiseptic, which is not an antiseptic, it's a disinfectant? Windex. Windex, if you read, you're not supposed to be putting on your skin, right? That's a no-no, right? Most of the things that you read, right, as we do our next project, tell you to wear what when disinfecting? Protective gear, gloves, maybe even eye protection. Not to get it on your skin, right? It's not an antiseptic. It's not safe for you, right? Make sense? Okay. And as I said, we're going to keep on this topic. All right. Pasteurization. This is brief heat treatment to reduce organisms that cause food spoilage and to kill pathogens. Surfaces can be pasteurized, right? So this is very common now. That good old Shark steamer that you can buy, right? That's pasteurization that you're doing. When you're steam cleaning, you're pasteurizing. You're not going to kill everything, right? But you're using heat. You're using a brief introduction of heat in the form of steam. So other terms that you'll hear are decontamination. And again, when we're trying to decontaminate something, we know that pathogens may be present. We want to get it to a level that will be safe. Degerming typically is a term that was sometimes used when we were talking about someone's skin, right? Again, we want to remove organisms that could potentially make us ill. Sanitize. This is an interesting one. Lots of people have sanitized buttons on their washing machines now, but most of us, if you're fortunate enough to have a dishwasher, you also have a sanitize button, right? Anyone know what that button means? It means that you just increased your electric bill probably by pushing it, because it increases the level of heat. And this is not a standardized level of cleaning, right? One manufacturer from another determines how much heat they consider sanitized. Do you need to push this button? Probably not, right? Unless you've got an infant, which it's not this class, my next class, I got 2 pregnant women right now, right? You don't need to push that button. Unless you have very young or very old, right, elderly individuals, you got grandma or grandpa living with you, right? You don't need stuff to be sanitized, right? A few of you guys, I don't have any vet students in this one, do I, in this class? No, it's my other class too, right? So their scrubs and stuff, after, you know, they've cleaned out the kennels, they might want to hit the sanitize button, right? Right? Those type of situations, you may need that extra level of cleaning. You may need that extra amount of heat, right? For most of us, under most daily activities, you do not need that extra level of heat or control, right? Your stuff is not that dirty. Make sense? Doesn't have to be that clean. All right, so save yourself a little money. Don't push that button. Okay, preservation. This is a process used to delay spoilage of perishable items. So this often includes adding inhibitors, ingredients, right, preservatives to our foods to extend their life. And basically what these preservatives are going to do is inhibit the growth of microorganisms that may be present, right? So they don't grow in our food. So why do we pasteurize milk? It's the main reason we pasteurize milk. You going to drink it straight out of the cow? Why not? It's good stuff. Bacteria are present, right, that could make you sick. Pathogens, right? That's the main reason we want to do it for milk. Are we worried about that with our wine? Are we worried about pathogens being present that are going to make us sick? No. We want the wine to last. We don't want it to spoil. So we're more concerned with microorganisms that are going to convert into things that we don't like, right? And that would be referred to as spoiling our wine. So to destroy pathogens is the reason for milk, right? We don't want to get sick. For wine, we want it to last longer. We want to preserve it. With our juices, so other than wine, right? Some of us are not wine drinkers. Juices are pasteurized too, right? Is it so we won't get sick? No. It's actually more so it won't spoil, right? So we can keep it on the shelf before you open it, right? And it's not going to spoil. It's not going to turn into something you don't want. It's not an animal source. Animal sources is where we have to worry about pathogens, y'all. Fruits, not an animal source, right? Unless some animal source has contaminated it, we shouldn't have to worry about pathogens. We're more worried about spoilage. Make sense? All right. So how do we determine how we're going to clean something, right? What are some factors that we need to consider? Well, the type of microbe, right? This is very important, right? Because when we started listing for chemicals, whether they could be high level or intermediate or low level cleaning, it was based on what organisms could they kill, right? What type of organisms can they destroy? The extent of the contamination, right? So in the microbiology lab, when one of my students dumps, and this happens every semester, happened yesterday, a broth culture on the desktop, right? It's full of millions upon billions of bacteria. I'm not like, just wipe it up with a paper towel and throw it away. I'm like, wipe it up, spray some Lysol on it, wait a couple of minutes, then wipe it up, right? Because you just dumped a whole bunch of bacteria on my desktop, right? So we've got to do more cleaning than you normally would do, right, than if it was, you know, some juice you spilt. The environmental conditions. So if I walked into my husband's room with some Windex and started spraying his Xbox, would I still be married? Probably not. Electronics and liquids do not mix, right? Not a good idea, right? These types of things you have to take into consideration. The risk of infection, right? When you're cleaning, you don't want to get yourself exposed, right? You want to protect yourself. So we've already, right, kind of dived into a little bit of this as we were doing our activity, right? So this is kind of review because I know I'm out of time. I will post this full one with all the answers, but most of the key stuff we've gone over. Really, your definitions are what's important. You do not have to memorize, right? Where is it? These definitions right here. Notice that the assessment of this one is to do a discussion, which right now we're working on that page in Canvas, and later on we're going to continue, as I said, we're going to do a discussion board. So this you do not have to memorize for test, right? Definitions before this that you need to know. All the questions in this you need to know. Okay? And I am going to post a recorded lecture from previous semester for you guys. So some of you guys that like to hear my little funny stories and stuff, it helps you remember this stuff, I am going to post that for you. So this is still content, right, that you need to cover on your own. It's easy stuff. I know you guys can handle it. Test is still Tuesday. Okay?